out of what just looks like background noise. That's important from a sonar viewpoint because when I emit something, it has spreading loss. So you can think of two primary sources of loss. There's spreading loss, which means I emit something at a point and it spreads outward. So spreading loss. Atmospheric attenuation has to do with absorption of the sound by the air, and it's frequency dependent. So sonar is not just distance, we're also interested in direction. As a human, we have our ears, right? And we have two of them. It's not enough to have two ears, we also have this structure around the ears, and that's called a pinna. I might be pronouncing it incorrectly, but that's the word. At least that's how it's spelled on the slide. Does anyone know what a head-related transfer function is? So I'll try to build some intuition. When there's a sound, so if I snap, right? When I do this at different points in space, what's received, you can't hear it, so I'm not going to do it that way, but when it's received in my eardrum, it changes, actually, depending on where it came from. You can measure this precisely and get what's called a head-related transfer function, which is a function, essentially, that given some point in space, and so by that, I mean there's azimuth and elevation. Given some point and the sound, I know what it looks like when it hits my eardrum. It turns out you can measure this for a person, and you can actually develop software that accounts for it, and so you can get surround sound, even better, with just two headphones. But you might wonder, and this is just a side note, why didn't that happen? Why do we have surround sound systems? Why is there not a big market for HRTFs? The answer is, it depends on you, what your HRTF is. If you have lots of piercings in your ear, it's going to be different for you. If you have essentially ear shape affects it, hair, head, shoulders, so all those things account for it. And you learn your own HRTF, you as the human, and mammals too. So that's why there hasn't been such a big market, because it's hard to find it and difficult to measure. So here are some more bats. This is the same bat, actually, and now this is the original picture that you saw at the beginning of the presentation, but now you see the bat is vocalizing at something. Does everyone see that its mouth is open? This is what you would see for this species if it was flying around. Actually, you would see it opening and closing its mouth as it flew, and like that, right? You wouldn't be able to hear something until you reach certain points of flight. We'll talk about that in a moment. So this bat is vocalizing at this thing that's covered in felt, black felt. Can everyone see it on the right side of the slide? It was trained to do that for an experiment, and so now this is just a for fun photo of it doing it. Now it's flying over it. It's looking also there. I'll take this moment to say, for those who don't know, bats' wings are their hands. In fact, Chiroptera, and my biology is weak, but I believe that means family, or that's the family organization for them, means wing hand. And in this photo, you can see the bat's hand fairly clearly. As in, you can see, I'll try to make my arm look like it. There's the elbow up to the hand. You can see the thumb, which you don't see any photos of this here. I can show you them after the talk. That's used by the bat to grip on things when it's walking or doing other tasks. And then the wings, of course, are built up by the digits extending out. And so the bat is not trying to have sex with this. It is trying to scoop up what it was trained to think of as a target. This bat has a tail membrane. Can everyone see the tail membrane? It should be more obvious in this photo. In the bottom left, you can see the bat's foot, the feet are surprisingly small, don't have much function in this species. And there's a tail, and there's skin in between. When this bat catches an insect, it's often the case that it's difficult to catch an insect in its mouth because its mouth is so small. So it catches insects by hitting them with the wings or scooping them up with the tail. In this case, it's trying to scoop up the target, which is why it's making that motion. So now I'll show you a brief video. In experimental context, when you want to study something, you want it to be controlled. You want to control for variation. And for bats, we have a room, we as in at this lab in Maryland, where the walls have padding to reduce echo, fairly standard issue for anyone who's doing acoustics work. We also have a video tracking system to track the position of the bat. That is, we need to know with fairly high resolution where the bat is in space as it's flying at any moment in time. So on the left, you should be able to see, there we go. There's a bat flying through a room. In fact, I'm in the bottom right portion of that raw video. Can everyone see the bat? And on the right is the position that's tracked by the video system. So the gray pillars are these artificial trees we set up. They're not really trees, they're just columns of net that the bat likes to avoid because when it runs into them, it gets tangled or otherwise doesn't feel good. The magenta shape is a net that you cannot see so easily from the raw video, but it's put up to prevent the bat from circling around the forest of artificial trees. So when we have this position information, then we can ask questions about what's happening at that point in space. That is, where the emitter is, that is the bat, as it's flying. This is an example vocalization by the bat, an echolocation call. We say it's an echolocation call as opposed to a social call because it's in a room with no other bats. This is the time waveform. That is, x-axis is time, horizontal axis is intensity, measured at a microphone on the floor. So when you look at this, you might try to make statements about the structure, but it's very difficult to, I claim. So here's a spectrogram of it. And now we can see the structure that was in that was not so obvious. Again, red corresponds to greater intensity, blue is lower. And the y-axis here is different than we saw before because it's ultrasonic range. So if it's difficult to read for you, I'll help you. The lower 1,0,0,0 is 10 kilohertz. And this plot goes up to about 92 kilohertz. And so there's what's called a fundamental sweep, which is the lower red structure. Again, horizontal axis is time. This is about 4 milliseconds. The call begins at about 68 kilohertz and sweeps down to just below 30 kilohertz. That's the fundamental. You can also see what's called a harmonic, which is the structure above it, above the fundamental. Is that clear to everyone? A harmonic, by definition, is a multiple in frequency of some other signal. So a harmonic of 100 hertz is 200 hertz. Harmonics are what make music beautiful, for example. Well, they contribute to the beauty of music. Tone frequencies are not so pleasant to hear. Anyway, this bed has a sweep. It's called a frequency sweep or a chirp. However, so we see that, but the shape of the chirp, the vocalization structure, depends on what the bat's doing. And so if we're interested in building little robots that can fly around and track something, we should know what the bat does when it's tracking. And the full tracking process is called an attack sequence. The call structure actually changes throughout the sequence. In this plot, which is taken from a paper, you can see different call shapes. So as we move from the top to the bottom, we're going essentially from search in the open field to just before attack, or rather just before capture. The numbers on the left, the large numbers, are milliseconds. It's duration of the call. Left column is time waveform. Center column is spectrogram. Right column is what's called just a spectrum, magnitude plot. It shows energy across frequency for the whole call. So the lesson of it, if you move down the middle column, you can see that the bat begins with not much frequency modulation. That is, the spectrogram, again, I don't have a pointer, so just, if it's unclear, interrupt me and I can change the presentation mode and point with the mouse. But in the middle column, the topmost plot is fairly flat, which means the bat is not modulating its frequency that much. And also, it's a fairly low frequency. So we can try to rationalize this or understand it by noting that and I didn't tell you this earlier, so you wouldn't note it, but now we're noting it. Atmospheric attenuation depends on frequency, and as you go up in frequency, the attenuation increases, which means the 65 kilohertz is going to be absorbed by the atmosphere a lot more than 30 kilohertz. And so that's one reason why we might suspect the bat has a fairly constant frequency emission at search, and it's long and constant, which means there's a lot of energy in a particular frequency. The argument there is it's easier to detect something. So if I'm in a room and I have nothing except a small sonar system, the best way to detect something, detection, as in I want to know if something's there, not necessarily where it is to send out as much energy as possible intuitively. And then if there's something there, whatever size it might happen to be, I'll get an echo back. Okay. As we move down, the call shape changes, moving down the middle column. We can see it gets shorter, and there begins to be more high frequency content, and the harmonics begin to appear. The argument for this is as the bat begins to find, or has found a target, and is now tracking it, approaching it, it knows that there is something there, and it's fairly close to it. And so what it's trying to do is find out where it is. It's trying to improve the resolution. So in sonar, that would correspond to increased frequency. So that's why we begin to see spectral content up to 60, 70 kilohertz. Yep. No, so interesting question, but the bat vocal structure is very similar, if not the same, as ours in basic structure. The harmonic, I don't know. I'm not sure that it is well understood, but if it is, I know which books to look in to try to answer that question, but I don't know. Because there are actually species of bats that can focus or defocus energy in different harmonics, so there might be something more. In this species, though, to my knowledge, they don't have that control. So, and then at the end, toward the bottom, the pulses are about 1.1 millisecond long. So that is to say, to give you some intuition, a 1 kilohertz tone has a period of 1 millisecond. So these entire calls span only 1 millisecond. So this is how quickly the bat's generating these calls as it's approaching something. And as there's more high frequency content, which is further expanded because of the harmonics, there's also lower frequency content. As in there's a shift, and I'll try to give you the intuition with my arm, in this direction. And if you stand near a bat when it's catching an insect, you can actually hear a buzz, which has a sound like this. It doesn't go up. Mine went up a bit, but it's called the terminal buzz. Before echolocation was discovered, that's what scientists would hear. But they didn't know how to explain it. Well, we'll get to hear a recording that's slowed down by a factor of 10. Yeah. We'll look at the recording in a bit. A few more slides. So neuroscience. We're not going to talk much about it, but the science of the nervous system. Bats are mammals, which means in many respects they're similar to us, which is why they're such an interesting model. Well, that's part of the reason why. They're similar, but they can fly. They echolocate. In fact, their primary system is audition. That is, whereas we in primates are vision heavy, and we have lots of parts of our nervous system that have visual responses, bats are different in that they have acoustic responses. But so more generally, what I come across, at least insofar as open source hardware and in the hacker community, there's more of a fixation on EEG and non-invasive techniques, which is understandable because in humans you generally cannot be invasive for recordings. By invasive I mean if I stick a wire in someone's head, that's invasive. But if I put a pad over the scalp, EEG, MEG, that's not invasive. The problem with these, what I call population recordings, is that you're picking up the activity of an extremely large number of neurons. And it isn't clear that that's the level at which computation happens. And most significant results in neuroscience happen in animals where you're actually doing these invasive recordings. As a side note, you can actually have invasive recordings in humans, typically in the context of epileptic